Okay, thank you, Subhasis. I'm Michel Orit. I'm chairing this second talk of the webinar this afternoon. And it's my great pleasure to introduce Professor Philipp Kukura from Oxford. So Philipp has done a large part of his studies in Oxford already in the past. He has studied there and got his Master of Science. Then he moved to UC Berkeley in California for his PhD with Richard Mattis, where he did femtochemistry, so short pauses studying molecules. And after that, he moved as a postdoc to the ETH Zurich at the time, where the group of Vahid and Dogdar was very active in nano-optics. And together with Vahid, he invented what is known as IceCat. I suppose that's he will tell us about that today. And since the end of his postdoc in 2010, he's back in Oxford, where he has accessed all the different steps, research fellow, associate professor, and since 2016, professor of chemistry. And at the same time, he's also founder of a company called Refine, who is also doing very interesting things. Philipp has received many awards, and in particular an ERC starting grant in 2014. And I mean, he's very active groups. He's doing a lot of very nice publications recently, and we are very much looking forward to his talk. So Philipp, please, I see that you shared your screen already. Please go ahead. We look forward to your talk.
I just realized I'm muted. You would think I figured this out by now.
We hear you perfectly. Go ahead.
Very good. Well, thank you for having me. It's actually quite interesting. I was just searching through my email, and I was supposed to be in Leiden, I think in the middle of March last year. And so early March, I think at some point I sent out the email saying, I'm not entirely sure this is the best time to come and travel. And of course, you know, we all know what happened after that. So at least digitally, thank you for having me. I thought I'd start today with the kind of diagram that drove me crazy when I was in high school as an undergraduate studying biology, which are these kind of diagrams that we draw to try and capture the mechanisms of biomolecular processes. So the example that I've shown here is protein degradation, right? So you have a protein which is folded in a happy, sometimes they misfold. And when they misfold, they can become a problem. And of course, you need some kind of mechanism in order to get rid of these misfolded proteins. And nature has solved that in various ways. One of them involves, you know, a series of enzymes, which really what they do is they tag this misfolded protein with ubiquitin. And then if you get the right number of ubiquitins and sequence of ubiquitins, then this misfolded protein will be identified by the proteasome and it'll get chewed up. And basically disappear. So it's a great way to get rid of proteins that you no longer want. And if you think about how we arrive at these kind of mechanisms, you know, in a very simplistic way, you could say, well, we always ask the same questions, right? So you need to know what do you have? You need to be able to tell that you have E1 and E2 and E3 in the proteasome and your substrate. You need to know how much you have, right? So you may start out with some of this stuff and you end up with none. You want to know how it changes as a function of time, because that kind of gives you the sequence of events. And then also very important, you need to understand what interacts with what, right? So, you know, this interaction here, this interaction here. And if you can answer these four questions, again, very, very simplistically, then you have a relatively general route to studying biomolecular mechanisms. And we've developed, you know, with lots and lots of methods, and I could spend hours and hours talking about all the methods that are out there that try and do exactly that. Mass measurement is one of them. And the extreme power of mass measurement on mass spectrometry is that it's unique, right? So protein A has a different mass than protein B. So without having to do anything else, you can identify and you can separate a species and complex mixture. If you want to complain, you'd say, well, it usually only works in the gas phase, and you know, for biomolecules, it could be problematic. Then you have fluorescence, and the big advantage there is that you can look at single molecules. So you can actually watch individual molecules do their job. It's dynamic. You can do it in cells if you want. And if you wanted to complain, you'd say, well, the problem I have is that I have to attach labels because most proteins are non-fluorescent, so I have to attach. And then finally, you have scattering, right? Which, again, the advantage is it works with everything. It's universal, right? And the huge thing here is that you can get atomic structure. So, to be provocative, you could say, well, I'm going to be greedy and I want everything, right? So I want to measure mass because it gives me information. I want to use light because it's dynamic and I can do single molecules, and I want to use light scattering because that makes it universal. I don't need labels. And then when you ask that question, why can't I do that? Well, the first answer is, or would have been some time ago, well, you can't see single proteins. And the reason why you can't see single proteins is because they're just too small. They just scatter too little light. Another way of looking at it is, is that really true, right? So you've all seen something like this. If you're colorblind like me, all you'll see is different intensities on top of the water. And if you're not colorblind, then you'll see different colors. And of course, the reason why you see different colors is because of interference. So what happens is you have an oil film on top of water. You have your sun, which shines white light onto the oil film, and some of it gets reflected at the oil-air interface, and some of it gets reflected at the oil-water interface. And depending on the path difference between these two, you'll get constructive or destructive interference, right? So you'll see a slightly different color depending on what the thickness of that oil film is. And in fact, you'll get a different color even if the difference in the thickness is only about 5 nanometers. So it's quite remarkable, right? If you think about it, you use the sun as a light source, you use your eyes as a detector, and you're just looking at at water with oil on it, and yet you can see features that are of the size of a single protein, 5 nanometers. And then you'll say, well, you're cheating, Kukura, because of course what you've done here is you've integrated the signal over lots of space, right? So yes, you can see a difference in thickness of 5 nanometers, but it's over centimeters or more. Well, then the answer could be fine. So I'm going to work harder to confine not just vertically, but also laterally. So if I use a microscope, if I use a lens, then maybe I can confine laterally to 200 nanometers. And then the question becomes, can I still see these kind of differences if I don't use the sun and I use a good light source like a laser, and if I don't use my eyes, but I use the best cameras and lots of analysis, etc. And that's essentially what these experiments do. You have an objective, you have a laser light source, and you have a camera detector. And the kind of earliest implementations appeared from the kind of the godfathers of confocal microscopy now 30 years ago, where they took a confocal microscope, looked at microtubules, and rather than look at the fluorescence, they just looked at the reflected light, and they could see these black lines. And it's quite remarkable when you do the calculation here, you look at a diffraction limit at the spot, you come to the conclusion that the detection limit in this microscope was about 300 tubulin monomers. Which I think was probably by a long way the most sensitive, label-free biosensor out there 30 years ago. But nobody thought about it that way. Nobody looked at that image that way. It just saw microtubules. And then, of course, Vahid's group, they were interested in small gold nanoparticles, and again using a very similar approach. They could go down to about 5 nanometers. So really very small nanoparticles. Now, really particles that are the size of a single protein. And I kind of came into that, and what we realized is that if you optimize all of this, and if somehow you manage to do the experiment perfectly, which is the dream sentence for a PI and the horror sentence for every student or postdoc working on the project, right? If you just do the experiment perfectly, you can see anything, because your signal to noise ratio here scales with the square root of the number of photons that you detect, which means if your signal is very small and you can't see it, well, you just put more light onto your sample, because a certain percentage always gets reflected and therefore will hit your detector. Or you integrate for longer, or both. But the trick is, no matter how small your signal is, at least in principle, if you can eliminate the background, you should be able to see single molecules. And sure enough, 10 years later, there's single molecule detection. So we kind of stumbled across this accidentally. In a way, we were looking at motoproteins walking along actin filaments, and then suddenly we realized that we didn't only see the proteins that we labeled, but we could also see proteins along the actin filament that we didn't label. And we analyzed the signal, and basically the one way that we convinced ourselves that these were really individual motor proteins was that the signal that we observed was exactly the size that we would expect given the mass of the protein. So we scaled from actin to myosin. And in Vahid's group, they looked at a whole bunch of different proteins. Again, they could see individual proteins. And in both cases, there was this realization, which kind of made sense that the signal that we observe should be proportional to the mass. So then you ask yourself the question, okay, how good is that relationship? And where does that come from, right? So the signal is proportional to the polarizability in this interferometric detection scheme. The polarizability scales with the volume and the optical properties. And of course, mass is equal to density times volume. And so if you look at, you can calculate, I mean, I write here refractive index. I'm not entirely sure that that's the correct term here, but essentially you can calculate the optical properties of proteins as a consequence of the amino acid content. And you can calculate something that corresponds to the volume, to the density. And both of these numbers vary by less than about 1%. So, and that kind of makes sense intuitively, right? So proteins are all made of the same stuff. They're not some of them are made out of metal, and others are made out of rock, and some are made out of something else, right? They're all made out of the same amino acid. So optical properties should all be the same. And they're all kind of the same size, the same structures, right? Beta helices, etc. So you don't have proteins that are 40 kilodalton that are 3 nanometers in diameter and others that are 30 nanometers in diameter. They all also have the same density. So at least in principle, it should be possible to really do these measurements quite accurately. One of the problems that we had, and this was certainly something that our referees of the original paper picked up, where they said, I mean, this is all very nice and well, but why on earth are you doing this? If you do single molecule fluorescence, your images just look so much better than this kind of crummy stuff that you can just about see there. And the challenge is somewhere here, right? You see this number here, differential contrast. So if you want to think about it in this way, when a protein is there, it changes the reflectivity of the surface by something like a fraction of 1% or less. So you somehow have to measure this change in reflectivity. And if you do the calculations, you essentially come to the conclusion that because of the way that detectors work and cameras work, it's really difficult. It's really quite tricky to reach this regime. And the reason why the signal is so small, of course, is the reason where we started, which is it's hard to see single molecules because they're small compared to the wavelength of light, and therefore they interact weakly. So however you do this, you have a lot of background light and very, very little scattered light. It's essentially captured in this equation here. So you have, in the case of iSCAT, where you're looking at reflection, you have a strong reflected term and a weak interference term. So right around the same time, we were also playing with dark field microscopy, and we found out that if you're really, really careful, you can actually be exquisitely sensitive, right? So you put a beam stop just behind a microscope objective, and we could see 10 nanometer gold particles, and we could even see the fluorescence from single dye molecules without actually having to use any filters, which tells you that you're suppressing your incident light by many, many, many orders of magnitude. And essentially what you're doing there is you're reducing this term here, right? You're reducing the background term. Now here you're measuring in transmission or in reflection, it doesn't really matter. The bottom line is you're changing the relative amount of scattered and background light. And apparently it's possible to reduce the background light towards the level that you would expect from individual molecules. So you take this lesson, you take our regular setup, the lessons that we learned from here, and the knowledge that single emitters at an interface, like glass-water interface, preferentially emit into certain directions or into high angles. And you just kind of adapt this setup where you scan the beam and then you look at the light that comes back to re-image the backfocal plane and put a little mask in. Then you should be able to increase the contrast, because what's happening is your scattered light is here mostly on the outside, whereas your reflected light from your laser is coming through the middle, so you can selectively attenuate your reflected light compared to your scattered light. And now you get images like this, right? So suddenly for a 400 kilodalton protein, we was really starting to see the kind of image quality that I dare to say even someone who does single molecule fluorescence would be quite proud of. And at the same time, as excited as we were about this, it wasn't quite enough. And why not? So across the hall from me is a colleague of mine called Justin Benesh, and he's a mass spectrometrist, and he uses mass spec to study protein function, malfunction, interactions, mechanisms. And of course, mass spec has huge levels of mass resolution. And he kept looking at what we had. And he said, that's just not good enough, right? And we were in this pub here in the Chester Arms in Oxford, and maybe had a few too many beers. And he convinced us that we should really try and see how far we can push the technology. And really, we did two things. So one was to very carefully look into the analysis as how do we actually quantify the signal from a single molecule, right? How accurately can we determine how big that contrast is? And the other one was to really work on the setups and try to design out vibrations. And essentially, the way this works is you have a background which is usually generated by your cover slip, and what you do is you average images at some time before and after an event. You subtract them from each other. If nothing happens, you get no signal. If at a point T1 a protein binds, then you will subtract your background with the protein from your background without the protein, and you will see your protein. And at a later time, your protein is already there, so if you do the subtraction, it will appear that it's gone. When you actually look at this movie, then the movie looks like this. So you get these black dots that appear and disappear, and the reason why they appear and disappear is not because the protein arrives and departs, it's because of this subtraction approach. And you can see the signal kind of grows in and disappears and grows in and disappears, and it does that in a very well-defined way. And if you take advantage of that, then you can very accurately determine the contrast of even one single protein. To give you an idea, this kind of baseline noise that we've been able to achieve, that's about equivalent to our substrate moving by an angstrom during the integration time. So if your substrate moves by more than an angstrom, then all you see is background due to the drifting of the cover slip. And it's really these two things that resulted in the development of what we call mass photometry, which is really the highly resolved and accurate measurement of mass, right? So here you now have a movie of BSA, and again, you see these signals, and you see sometimes they're quite faint and sometimes they're really quite strong. And if you quantify every single one of these events, depending on how strong the event is, it will be along this axis here. And depending on when it lands, it'll be along the time axis here. And then you see these dots, and immediately you see there's a band here, and there's another band here. And then when you integrate up, you'll see there's another band here, and maybe even a fourth one there. And the reason why you have 4 bands, and all these 4 bands are equally spaced, is because it's a monomer, a dimer, a trimer, and a tetramer of BSA, right? It's the mass of 1, the mass of 2, the mass of 3, the mass of 4. And the key thing here is that you can tell the difference between a monomer and a dimer, or if you like, the difference between 2 species being separate and 2 species having formed a complex. It's really the resolution that is absolutely key. The second thing is, because we can measure the contrast now so accurately, that translates into very high math accuracy. So here's a whole bunch of proteins, and by now there's several hundred, if not thousands, and the accuracy is about 2%. Now, if you do mass spec, you say, well, that's maybe not quite good enough. But actually, for many applications, that's plenty for what you need, right? Because if you think about it, what I'm saying is, if you have 150 kilodalton antibody, that means that on average, we will measure between 153, is my math correct? Yeah, between 153 and 147. That's usually good enough to know that what you're looking at is an antibody, right? Versus maybe something that the antibody binds to, which might be at 95 kilodalton. And that's really gives you this unique capability of mass spec, where you now, at least in principle, don't need anything else in order to be able to identify different species in a mixture. Since then, we've spent quite a lot of time and effort in finding out how well this actually works, right? So this is all well and good, highly purified samples, and you get specific protein, and how well can you actually make this work, and how broadly applicable is it? So we've looked at self-assembly, where the question is, how do you go from solubilized proteins to some kind of assembled structure? In this case, it was kind of artificial nanocages where we could actually monitor the assembly as a function of time. We've become very interested in membrane proteins. Of course, membrane proteins immensely important because they're drug targets, but also immensely annoying because they want to be solubilized. So that makes them more heterogeneous. It makes them more complicated. They're attractive as targets, but very painful to analyze. And then also we showed that what we do with globular proteins also works with nucleic acids. And I still can't tell to this day whether that was completely obvious or not, because of course the polarizability, the description of the polarizability involves bulk properties, right? Whereas now you really have a molecular wire, right, which is surrounded by solution, and still, you get this very nice kind of linear behavior. And the contrast is very, very similar per mass as we've seen to proteins. Not quite the same, but similar. All right, so today I just want to mention three things, talk about three things, two of which are published properly, if you like, and one of them which is on preprint. One of them is how you can use the technology to look at protein-protein interactions and kinetics. Second, how it might help you in structure determination, which of course is of immense importance. And then finally, to show you some recent results where we've kind of moved away from proteins binding to a surface and rather have them move around. So in terms of interactions, this is a sketch that was not done, but dreamt up by the graduate student who was leading this work. If you think about affinities, right, and the equilibria, does A bind to B, right? Do they interact or not? What you're really interested in is how much of the complex do you have compared to how much of the free species. That's what you do. And normally you express that as concentrations, and that's what gives you your binding affinity. So, usually you will measure the concentrations or you will measure rate constants, and from that you get your affinity. But if you can actually see single molecules and you can measure their mass, then in the complex mixture, you can count how many free antibodies you have, and you can count how many antibodies have the antigen bound, and you can count how many free antigens you have. And then you should be able to directly determine binding affinity. So here's a super simple example. So tubulin. Tubulin forms a dimer, and if you look into the literature, you'll find the affinity for dimerization to vary by 4 to 5 orders of magnitude, depending on what technique people have used. Right? If you take tubulin and you dilute it down to 20 nanomolar, you get 2 peaks. You see one here, one at around 100 kilodalton, one around 50 kilodalton. That's the amount of tubulin. And if you integrate how many, count how many peak particles you have here and count how many particles you have here, and you plug them into the equation, you get an affinity of about 8 nanomolar. Okay, so that's the dimerization affinity. And then you say, okay, is that really it, right? And am I right or not? So the natural thing to do is then to do a titration. So you go from low total concentration to high total concentration. And if we're right, as you go across the binding affinity that you've measured, what you should see is a transition from mostly monomeric species to mostly dimeric species. And that's indeed what you see. And at each of these concentrations, you can count how many you have in both and try and determine the KD with a single shot approach. And you see that, again, you get roughly the same number. Or if you don't trust that approach, you can do the kind of more traditional fitting. And again, you'll find that you can extract these numbers really quite accurately. The other way to get affinities is through kinetics. So here you have, antibody-antigen interaction. So this is something that a lot of people are interested in. And we've taken an antibody, and then we've taken the same antibody and taken the sugars off. In the presence of the sugars, you can see you get this very tight binding affinity, about 50 picomolar. And the way you see that is, here's your free antibody, here's your free Fc gamma. When you mix them together, even at low concentration, you get almost no free antibody left, and everything is complex. So that tells you that the interaction is very strong. If you have the deglycosylated antibody, you do the same thing at the same concentration, you can still see a good amount of free antibody and only some complex. And again, you can use the same approach as before in order to determine the binding affinity. The other thing you can do is you can do what you do in SPR, where rather than measuring equilibrium distribution, you measure kinetics. So if I'm at a high concentration, I dilute down, how long does it take for stuff to fall apart? Or if I mix things together, how long does it take for complexes to form? And from these kinetic curves, again, you can get on rates and off rates, and you can also determine the affinities. And the reason why that's important is because people generally care about tight binding, but they also care about the associated kinetics, right? So the affinity is one part of the equation. The kinetics are another part of the equation. The second thing I promised you was structure, right? So here's my very skilled sketch of the proteasome. If you think about it, if you want to know whether your proteasome is intact, one way to do that would be to measure the mass. Because this thing has a very specific mass. If anything has fallen off, the cap or individual proteins are missing, you would get a different mass than the one that you would expect for the fully assembled species. And having assembled samples is important, generally, right? If you're running an assay, you want to see activity, you want to make sure that your machine actually is in the shape where it can perform its job rather than having fallen apart. But if you do structural biology, it's the same thing. Even though cryo-EM, it's unbelievably powerful, and presumably, soon enough this will no longer be an issue. But even for cryo-EM, the way you get a structure is you have to identify lots of individual particles, and then you have to average them together. And the averaging is the one that really gives you the resolution that you need in the end. What that means is that what you put into your 5 million pound cryo-EM, you want to be sure that your sample is mostly in the state that you need for structure determination, right? And so if you have lots of different stuff, it can become quite tricky to do this. So how does this work? So we looked at this rather large membrane complex here, and at the concentration which we measured, we saw fully assembled stuff, which is quite heavy, around 800 kilodalton, and then this little bit missing, which is over here, and then that bit all by itself. And you can see these different species in the mass distribution. And take exactly the same sample and you do a negative stain. Again, you can find examples of exactly these species, right? So here's fully assembled, which is this one. Here's the bit missing, which is this one. And then there's only a little bit, which is this one. And these are the corresponding mass photometry images, if you like. It's not exactly the same sample, of course. It's just stuff that we've taken out. One thing that I quite like about this is, if you look at the single particle contrast you get in an electron microscope, and admittedly limited by dosage and shot noise, etc., and you look at the single particle contrast you get with light, it's actually pretty good, right? I find it quite remarkable. Your wavelength is so much longer. You're just looking at one molecule, and you can still get very, very significant contrast. So what that tells you is that indeed the mass, or measuring the distribution of mass for a solution, will give you a very good idea of the composition of your sample. You can turn this around and say, well, often the particular structure that people are interested in isn't really stable under normal conditions. So they have to play other tricks in order to make it stable. They have to bind it to something, or another trick would be to do chemical cross-linking, something like glutaraldehyde. Where you're at high concentration, you mix in your cross-linker, you let it sit for some time, and you quench, and then you hope everything, you have a high fraction of assembled complex. And that's a reasonably laborious process, right? So we did exactly this here. Our collaborators were interested in complex number 3, and if you just looked at it under native conditions, so your complex number 3, relatively low proportion. But if you added 5 minutes of did 5 minutes of 0.1% glutaraldehyde, you see that this peak goes up. The other thing you notice is that it shifts to higher mass, and that's all the glutaraldehyde that you've added when you've actually cross-linked the sample. And this is just one example, but now you can see how relatively quickly you can scan different conditions in order to optimize your sample preparation. We then went all out and kind of went through a whole purification procedure as one would normally do, right? So you have affinity, ion exchange, size exclusion, And what people usually do is then they get different fractions from all of these columns, and you kind of look at these and you try and figure out where is it assembled. And often you will then do negative stain, and from the negative stain, when you kind of get the right structure, you go, okay, fine. If in the negative stain my dominant species is the one that looks assembled, then my sample purification is successful. So we went through this whole thing. So this is the mass distributions that we measure, right? And you see after step 1, at 1.2 megadalton, this is where we have the assembled complex. This is what you want. So initial purification, not so good. Then you do ion exchange. This fraction also not so good. But this fraction now, suddenly you have lots of assembled stuff. Okay, very good. Then you take that and you do a size exclusion chromatography. And again, depending on which fractions you choose, here are good fractions, here are bad fractions, right? And then our collaborator found some samples in the freezer that they produced in the process of 2 or 3 years of work, adding compounds and cross-linking and everything that was super, super purified, that gave them the structure. And sure enough, when you stick them in, you get, just a single peak for the assembled structure. And we took all of these samples and we did negative stain, and then we looked at the top 4 classes. So what is the most abundant species? And sure enough, you can see that where we saw a large proportion of species as 1.2 megadalton, that's also where in your electron microscope you predominantly saw the desired complex, if you like. So where it helps is, well, it's very quick and easy to do, right? And it's very quick and easy to scan different conditions, and you can kind of get the full distribution of stuff. The other thing where it helps is we really only measure mass, and for the negative stain approach to work, you have to have a pretty good idea of what you're looking for, right? So if you look at something like cohesin, which is something that people are trying to get the structure of, which is much more floppy, then in your negative stain, things look really quite messy. It's very difficult to classify. At least the mass photometry will tell you that your sample is really very pure. It might just not be structurally pure enough to determine for further characterization. And next one I want to talk a little bit about, I think am I still doing okay with time, Michelle?
Yes, I think you have something like 4 to 5 minutes.
Okay. Is membrane proteins, right? So the problem with membrane proteins is they like being in membranes. So if you want to characterize interactions, what you have to do is generally you have to take them out of the membranes. Most general approach is to use detergents. There's more complex, more gentle ways, but generally it's detergents. And of course, if you have detergents, the problem is if this is surrounded by micelle and this is surrounded by micelle, how are you going to understand the interactions between these proteins? Because what you're going to get is the interactions between the micelles. What you really want is, ideally, you want to look at these interactions in a bilayer, right? So in principle, in our normal assays, we have proteins binding and unbinding, and all we need is presence versus absence of a species. You can also achieve that by proteins moving. So imagine that you have your protein sitting here on the surface of a bilayer or inserted in the bilayer and they diffuse around. And essentially, that's what we did, and this is the kind of movies that you get. So dynamin 1 is a membrane-associated protein. It's not a transmembrane protein. It binds, if you have the correct charge lipids, it binds to the surface of the membrane and moves around. And now you see kind of a very similar thing to what you saw before, except now you don't see proteins arrive and disappear. Now you see them arrive, move around, and then disappear. And they arrive when they bind, and they disappear when they unbind. And you also see that some of them are really quite dark, and some of them are really quite faint. Do the same analysis as before. Right? You can get these, regular spaced intervals. You can calibrate, so you get the mass, so you know that this is dimer, tetramer, hexamer, octamer. The localization precision at 300 hertz imaging speed, which is what we use, is on the order of few to 10 nanometers, which is actually quite comparable to what you do with fluorescence. But of course, what you can do at the same time is you can measure mass. Right? So not only do you know that something is there, and you really know when it's gone, so it's not bleaching or anything else. You can measure the mass. You can get very long trajectories. You can determine diffusion coefficients. And in this particular case, now because you can measure mass and diffusion coefficient, here's a 2D plot of mass versus diffusion coefficient. You can see this, the dimers are quite fast, the tetramers are slower, hexamers even slower, octamers even slower. And then plot the diffusion coefficient versus 1 over the number of subunits, you get a straight line. You can also determine the residence time on the surface, residence time versus 1 over the number of subunits, again, a straight line. And what that really tells you is, a dimer has 2 contacts, a tetramer has 4 contacts, hexamer has 6 contacts, octamer has 8 contacts. That's when you would expect these properties to scale in this way. So you're now in a position where you can study the behavior of these proteins as a function of what complex they have formed. In this case, it's a homo-oligomer, so it's trivial, but in the future it could be more complex. And sometimes we saw events like this. So you see this big thing here diffusing around, kind of jiggling, and then it separates, right? It separates into 2 subcomplexes. Or here, you have 2 that are moving around, and you wait long enough, and they join forces and they become 1. And for me, that's the kind of glimpse, right, into the future where you can start to study protein-protein interactions of membrane proteins in a bilayer, which is really, really quite difficult to do. So I hope I've convinced you that shining light on proteins and thereby measuring their mass, you can do lots of interesting and useful things with it. I think one thing I would just want to end with is, if you look at EM, right, over the last 15 years, there's a kind of resolution revolution where, 15 years ago we were here, and now atomic detail, you can see hydrogens, etc. The way this happened was, better detectors, better lights or electron sources, lots and lots and lots of data analysis, and better sample preparation. And at least in my mind, I don't see any reason why we shouldn't be able to achieve something similar here, right? So this is the kind of resolution. If we take a full mixture of proteasome, this is the kind of resolution we had 2 years ago. Instrumentally, we are now getting pretty close to what you see here in the middle. And I think, if you work on the detectors and the optics and the data analysis sample preparation, I don't think it is unfeasible that you can get over here. And the reason why I think it's significant is because it takes me all the way back to where I started. If you can measure mass, you can see individual molecules, you can measure mass accurately, you can achieve this very, very high resolution. You can see all of this stuff, and you can see how it changes as a function of time, and you can distinguish different species. So you have a direct route to study biomolecular processes when everything's okay, also when you have malfunction, and you can also start to look at what happens when you try and intervene. So, I think the future here is really extremely exciting. And with that, I'm done. Want to thank the people who actually do the work. There's a recent photo of my group, because I just say, in principle, your signal to noise ratio is unlimited, so I just keep bashing away, it'll be fine. I want to say especially thank you to Justin as well, who's had a monumental input on the development of this technology. And Michelle, thank you again for having me, and I'll try and answer questions.
Thank you very much, Philipp, for this very inspiring talk with many exciting and huge perspective in applications and new science. So please ask your questions either directly or by the chat. And I see already one question by, I think, Subhasis. Yes, Subhasis, go ahead, please.
Yeah, thank you very much for your nice talk on the different applications based on the iSCAD. I was thinking about in the beginning of your looking at beginning some slides about the diffusion, and which you covered at the end of your talk, but maybe I will still ask that question. So one thing what I see that you get the if you have just normal glass surface, you don't have any signal, and then protein binds, so you get interference, which is you see the destructive interference, and then you get that signal. But this interference will depends on the where the source of your scattering. So if it's much close to the glass surface, or if it's away from the glass surface, so that will change the interference signal, which can be related to the diffusion or 3D diffusion, and can be to track that things. But what mostly in the beginning I was looking at the movie where I see mostly it's a dark spot. It's mostly the destructive interference. There is not much thing happening. And then there is one point also comes, it's just an image subtraction between two frames. So which means your dynamics and the image frame integrations will really depend. So if there is a fast dynamics, then the just molecules move away in the one frame, so you don't have much information. So that is really related. So I wanted to hear from you about this point. And I have a quick second question I just ask now about the beam blocker, which you use to reduce the reflection. And that is basically the reducing the reflection from the glass surface. So that is one kind of reflection source. But there can be many other kind of reflection source, like reflection from the objective. And do you have any problems to reduce that is also? Yeah, thanks a lot.
Yeah, so both points very well made. So for the first one, you're absolutely correct. And the honest answer is we don't see any dynamics in those kind of assays. And the reason is that proteins, as you saw in the previous talk, right, they diffuse very, very quickly. And we need a lot of time to integrate enough photons to get our shot noise down to be able to actually see the molecule. So the only molecules that produce signal for us are the ones that actually get immobilized. So, you know, it's really amazing to see that you can now detect single molecules, you know, in microseconds, right? And of course, if you can do it in microseconds, then you really start to get information. You can start to try and disentangle what happens as the molecule is moving closer or further away from your, you know, detection area, if you like. We can't do that. We just, you know, we really, the events that we see are the ones where, you know, it sticks. And because we integrate for 20, 30, 50, 100 milliseconds, and we compare 100 milliseconds before and after, right? The only events that we see are events that are unchanged on that time scale. And unchanged on that time scale is non-specific absorption, irreversible absorption to the surface, or irreversible detachment on the surface. With respect to your question on the mask, you're 100% right. That's the reason why you can't play this game forever. So, I mean, in principle, you can, right? So you could do this in dark field, and in fact, you're going to see something. But the problem is, the stronger you make this mask, the more background light you get. And you're absolutely right. It's mostly background light from the objective, from spurious scatterers, from reflections, from, you know, anywhere. And of course, that light you don't want, because that light is not interferometric. It does not produce contrast. It maybe changes as a function of time. It's uneven. It's just a problem. So we are just kind of cheating a little bit, right? Your strongest light source on your camera is from the reflection. And we just attenuate it a little bit. And it doesn't change this, you know, the nominal signal to noise ratio does not get better. You don't, you know, but the reality of it is, you know, we have, I don't know how many setups. We don't have a single one without them, because it's really difficult to find focus, and it's really difficult to make the measurement repeatable. And it's really difficult to really get these, you know, the monomer dimer thing, which is pretty much every measurement that is meaningful that we do. Like without it, we can't make it work.
Yeah, thank you very much.
Okay, because I don't see any immediate question, I get a chance to ask a short question, which is maybe a bit crazy, but you will tell me. So in principle, when you have a polarized object in a field, you get depolarizing fields. And I would expect to have a little component of this field, you know, influencing the scattered intensity. Is that completely negligible? I have no idea. I never did the calculation, so I have no idea of how big this effect is. And is that maybe responsible for this 2% deviation you have, or are those just fluctuations of the density index or refraction of the proteins that nevertheless exist, right?
So to your first question is, I have no idea. Unfortunately, I really just don't know. And to your second question, unfortunately, I also have absolutely no idea what the sources of the deviations. Well, you could see, if you look at the plot, it gets worse as you get to low mass, right? It becomes, the scatter gets more. one of the issues that we have is we don't really know what the truth is, right? Because our conversion from contrast to mass is empirical. We measure a whole bunch of proteins, we get a line. This is the conversion. And then the question is, okay, which protein is on the line and which protein is off the line? And why is which protein on the line and which protein is off the line? And of course, the next problem is the accuracy, this 2%, that's really close to our measurement error.
Yeah.
Right.
But I could imagine that you could see, for example, a change in shape of the protein, for example, when unfolding. Or
so interestingly,
in principle, this should be there, the signals.
So there are some studies out there. I think people have done this where they looked at, they actually added urea and denatured the proteins. And the main effect that people saw was something used to be a dimer and now it's a monomer. There wasn't a very significant shift in the contrast. It was probably a small shift, but then you never know, is this small shift because I've got my protein unfolded, or is this small shift because I slightly changed the conditions? And so there's this question mark. So we've been playing with it. The answer is, I'm pretty sure you are right, but the effect is small. And at the current levels of precision, we find it hard to measure. And maybe to jump into our defense a little bit, right? You know, the nominal signal to noise ratio at 400 kilodalton is now close to 100 for one protein, one molecule, right? So if you think about it, you know, if you do super resolution fluorescence localization, you know, below 5 nanometers, it becomes a nightmare. You have to correct for everything, orientation, camera, gain, like it just becomes a nightmare. To maintain that level of precision or accuracy on the determining of the contrast, you are in the equivalent realm of well sub-nanometer localization of fluorescence. And we haven't quite, you know, we haven't been able to push the analysis to that degree. I don't think that is fundamentally impossible, but in order to be able to answer these questions, we're just going to have to become, you know, I think an order of magnitude better than we are today.
Great. So that means further challenge for the future.
Yeah.
I see another question. So please, Abhijit Lavanya, please ask your question.
Thank you so much for that great talk. I had a question about the dynamics that you were measuring, and if you have to correct for the fact that you might have differently weighted things that arrive on your measuring surface at different times. Yeah,
you're absolutely correct. It's a very good point. It's something that we were worried about, and we still always worry about. So you can actually measure it. So we can do an assay where essentially we have a relatively high surface to volume ratio, and then what will happen? So this is your standard flow cell, really, with 2 pieces of cover glass. What will happen is at the beginning of the measurement, you'll have quite high, or you have your starting protein concentration, and then as proteins stick irreversibly to the surface, your concentration in your solution will drop. And as your concentration in solution drops, that means the frequency of collisions with the surface also drops. Because we can measure, we can see differentiate between different species by mass, we can look at the drop in collision events for an antibody compared to the antigen, or for an antibody compared to the complex. So if there is a huge difference, right, one is super sticky and the other one is not, we can tell. It turns out, and again, this is something where I think we've just been very lucky, because the adsorption to glass is non-specific, everything kind of sticks pretty much in the same way. You can measure it, you can correct, and then, you know, you go from here to here, but, you know, the influence, what you're actually seeing is very, very little. So we usually don't bother. But by and large, the differences you see scale with the cube root of mass, which is what you would expect from diffusion. And then you think cube root of, you know, even a factor of 3 in mass is big. It's not that big an effect. And then you just do this and you recalculate the binding affinity, and it makes almost no difference. Conclusion being, you're right. It's something one needs to be aware of. But so far, it hasn't been a massive effect.
Great. Thank you.
Thank you. If you have a question, please ask. I don't see any question in the chat at the moment.
This is Kobayashi.
Oh, there is one. Yes, please. Lisa Maria Needham, please.
Hi, sorry for my dodgy video.
Go ahead. Yep, go ahead.
Hi, I have a quick question, Philip. Can you get any more information, say, on say the orientation or the 3D spatial information by point function engineering, say placing a phase mask in your emission path?
I don't know. I think it's a very good idea, but the answer is I don't know. I mean, I'll be honest with you. My very, very naive, not very well scientifically founded, hand wavy argument always has been proteins are really small, and the differences in structure that you get, let's say, between an antibody that looks like a Y and a globular protein, because there's no delocalization of electron motion, are invisible to visible light. You know, if you have a gold nanorod rod versus a sphere, that's going to make a big difference, but that's because, you know, electrons are actually moving across the entire structure. But if you have a protein, it's just amino acids that are just sitting there in space. And at least to me, it's not immediately obvious that you would get a significant effect. I would say my answer to you is probably approaches the one that I gave Michelle, which is I'm pretty sure that there is an effect, but from experience, it seems to be small. And at the moment, it seems to be small enough that it's just painful enough to measure. And I would expect that you'd see a similar effect with PSF engineering. That is not to say that there isn't lots of interesting things I think one could or should do with PSF engineering in order to optimize the sensitivity and the information content that you can get. I think there's a huge amount to be done. It's just not really something we are experts at, so we haven't really implemented it. But if you think about it, right, the reason why the mask works is because we're taking advantage of the fact that the light is coming in differently from the light that is coming out. And we've implemented the simplest possible version of that. You know, I'm sure that there are better ways of doing it, and maybe you can get more contrast and more sensitivity.
Thank you.
Okay, thank you. So we take a last question from Professor Kobayashi.
I have two questions. One is, I enjoyed your talk very much. And the first one is, you mentioned that 10 kilodalton is baseline noise. And is there any way to reduce this baseline? And also another one is your system in terms of photon noise. Is it already close to the shot noise limit?
Yeah, so those are very good questions. So the 10 kilodalton is kind of very hand wavy. I mean, you saw we get these plots where we get the size of the signal versus mass. So we can convert that and we get a number that says, I don't know, 4 times. Sorry?
How much better you can expect in near future?
How much better?
Yeah.
Oh, I'm going to get in trouble here. So I don't think that 1 is unreasonable. I think technologically that should be achievable. Whether you will encounter other sources of noise that we don't know about yet, as you become more and more sensitive, I'm not sure. But you know, I'm thinking that, for example, the flyby detection should give you a pretty good idea of what, you know, for example, intrinsic fluctuations are like. So it makes me hopeful that that's possible, but at the moment we don't have an instrument that can go that far. So I will really only be able to answer the question whether it can be done when we have that. I think technologically, I'm convinced that 1 is possible because that's a back of the envelope calculation. Whether you can realize it in real samples, that's another issue, another question.
And your system is in terms of photon?
Yes. How close? I mean, it depends. When we do our experiments, so the movies that you will have seen, we're usually within a factor of 2 of the shot noise limit, roughly. But what you haven't seen is I haven't shown you, I haven't shown you a movie of 5 kilodalton proteins, and you'd say, but you told me that your signal to noise ratio is proportional to the square root of n. So why don't you just integrate longer and you see 5 kilodalton proteins? And the answer is, you don't stay shot noise limited forever. So if you integrate for a long time, you get other noise sources and drift that come in. And if you integrate for a short time, there's only so much power you can put on. So at the moment, that's the reason why it's roughly around 10 kilovolts.
Okay, thank you very much. I enjoyed your talk very much.
Thank you very much.
Okay, thank you very much, Professor Kukura. I think in view of the time, we have to stop the discussion here. I thank you everyone for being here and asking questions and for the discussion. And I announced the next webinar on photothermal microscopy next week with Professor Silvan Schmidt from Vienna and Professor Randall Goldsmith from Madison, Wisconsin. So I hope to have you all at the webinar next week and wish you a good week in between. Goodbye.